everyone. Thank you so much for joining us for our SevenBridges webinar. So my name is Xavier. I'm a Scientific Engagement Manager. So together with me is my dear colleague, Vladimir, who is a very knowledgeable bioinformatics analyst specializing in neoantigen discovery. So in this webinar, we are very excited to share with you some of our very new neoantigen discovery workflows, which will enable you to accurately predict your immuno-oncology targets. Next slide, please. So in this webinar, we will start with an overview of our company, SevenBridges, not only who we are, what we are known for, but also how we can make your research life more productive and hassle-free. Then I will switch gear to the neoantigen discovery, where I will tell you some background stories about neoantigen. So essentially what they are and why researchers are so fascinated by them. Then I will transition to Vladimir, who will elaborate more on the latest advances in cancer immunotherapy using neoantigen. He will share with us the successful story in neoantigen discovery and to showcase some of the very new workflows in that context. And after that, we will open the floor for a Q&A. So if you have any questions, please type them into the chat box below, and we'll respond accordingly. And I hope that by the end of this webinar, you will recognize and appreciate the full capability and the capacity of our platform to make your project from mission impossible to possible and hassle-free. Next slide, please. So who are we? So SevenBridges is a global biomedical data company specializing in software and data analytics to accelerate scientific discovery for our partners, both in academia and industry. We're a over 10-year trusted brand with a strong track record in delivering end-to-end bioinformatics solutions to our users around the globe. With us, you will have full access to our industry-leading bioinformatics platform, personalized scientific and bioinformatics services, transformative analytic approaches, as well as the largest federated genomics data sets. And with all these in place, we help you to advance your research path from raw experimental data to insightful discovery, which can lead to innovative treatments or diagnostics. And over the years, our credentials and capability has been well established by the community, as evident from all those awards that we have been receiving. Next slide, please. So for every single researcher, including myself, we all wish that our research journey can be a very simple, straightforward one-way street, where you start with an actionable hypothesis, then you do your experiments or analysis, then right off the bat, you have your novel biomarkers identified, which in turn advance your clinical study or drug development. However, in reality, it's not the case. Oftentimes, we have to go through iterations of testing and retesting of your hypothesis, which is very daunting and ineffective. So our goal here is to shorten your time and effort going through this iterative cycle in which every single researcher has to go through. And we can help you to reach your research goal as quickly as possible. As a result, you can move on to your next phase of your research project with full confidence. And to achieve this, there are three key successful elements that we will provide. First, the data set. Apart from your own proprietary data set, we also host multiple key signature public data sets. Next slide, please. So over the years, one of the top asks from the research community is to have a very simple and effective way to access any public data sets of their interest. So with that, SevenBridges have invested resources and efforts to build multiple key signature public projects, and we make these public data sets readily available and easily accessible by our community. Examples of these public projects are the CCLE, the Cancer Cell Line Encyclopedia, which is a joint effort among the Broad Institutes, Novartis Institutes, as well as the Genomics Institutes of Novartis Foundation, aiming to characterize close to 1,000 cancer cell lines. Another great resource for the cancer community is the TCGA, the Cancer Genome Atlas. This is a collaborative effort between the NCI and the NHGRI in profiling over 20,000 primary cancers across 33 cancer types. We also have the Human Cell Atlas, which is a great resource for a single cell study as well. Apart from the genomics data, we also support other omics and clinical data sets such as CBTAG, as well as the Cancer Imaging Archive. Next slide, please. And the second key element that will make you successful is our tools and workflows. In parallel to bring your own proprietary pipelines or applications, we also provide a collection of public tools and workflows. Next slide, please. So right on our platform, our public apps gallery have hosted over 400 gold standard tools and workflows, which are ready for you to plug and play. To date, there are over 100 individual toolkits spanning across 60 plus applications, which includes RNA-seq, RAS analysis, whole genome sequencing, as well as single-cell study. And this public app gallery is curated, maintained, and kept expanding by our bioinformatics team. It is highly optimized for the cloud, fully extendable, and customizable to meet your specific needs. On top of that, you can also customize any of these public tools and workflows using our web composer. So our vision here is to help our users, regardless of their programming experience, to succeed in their projects. So with that, we have developed a very intuitive GUI-based web composer, which allows users to customize tools and workflows by simply dragging and dropping, as shown in this animation here. So with that, you don't need to worry about typing pages of code. Instead, our platform will generate the code for you on the fly. And for every single parameter, you can set it to be exposed to users or locked, so that you can have a full control on the accessibility of the tool or workflow. And on top of that, every single version of the tools can be saved and retrievable right on our platform. This enables you to run your project using the same version of the tools every single time. And all these tools are wrapped in Docker containers and CWL, the common workflow language. This makes the tool very portable, shareable, and reproducible. Next slide, please. So for the power user, you can also easily bring in your tools and workflow to our platform using our SDK or software development kit. Along with the Docker and CWL, it will provide you remarkable portability, reproducibility, and scalability. This makes your tool migration in and out of a platform a very seamless and hassle-free process. Likewise, our version control feature will enable you to save and retrieve all versions of your tools right on our platform. So running the tool today should get you the same result versus another individual running it someday down the road. Next slide, please. On top of that, our platform is established based on the FAIR principle, which stands for Findable, Accessible, Interoperable, and Reproducible. We achieve this vision by engaging with the community standard organizations. Historically, CWL is founded by us and our partners. Since the inception, it has been widely adopted by over 30 academic and industrial partners, which includes NCI as well as GA4GH, to maintain the standard data formats and databases for genomics. Quoted from the biocompute object specifications, our workflows along with the CWL are dubbed as the appropriate ways to exchange workflows. Next slide, please. And the third key element which sets us apart in supporting our users is our well-established cloud-based infrastructure, along with our strong scientific and bioinformatics services. We are not just a platform company, but we are a strong team of over 200 engineers, scientists, as well as bioinformaticians located in different time zones. With that, we have the full capability to support our users around the globe and to ensure that their time-sensitive projects can run robustly right on our platform. This is in particular very important for any large-scale and complex clinical processing, such as neoantigen. Next slide, please. And in this webinar, we'll have neoantigen as our use case to demonstrate our platform capability. As a brief intro, neoantigens are essentially peptides that are presented specifically on the surface of the cancer cell. And this neoantigen can trigger an immune system where T cells with the right receptors can bind to the neoantigens and elicit the killing process of the cancer cells. And this cancer killing process, using your own immune system, is called immunity-induced tumor regression, or immunotherapy in short. In contrast to the chemotherapy, which is a standard cancer regimen, immunotherapy offers a very high specificity, which only kills the cancer cells and leaves the normal cells intact. So with that, leveraging neoantigens for guided treatment has emerged to be the definition of personalized cancer immunotherapy. And that's the reason why researchers are so fascinated by neoantigen. Next slide, please. And of course, any fascinating project will have its own challenges. Neoantigen in particular is a very challenging project in which they are a very unique entity among individuals. So in a clinical study with a very large cohort of patients, you will anticipate a very large and complex data sets to manage. Not only do you have to have the capability to process them, but also you have to process them fast enough in the clinical setting. Apart from the population level, each analysis per patient can also be very complex. This is due to the fact that we need to capture as many features or mutational events as possible to improve the accuracy in the prediction of neoantigen. And these features will include variances in somatic, germline, RNA levels, as well as RNA expression and HLA typing. All these combined take a significant amount of development efforts and resources. Currently, there are no best practices workflow in place for neoantigen analysis. There's also no true set available for evaluating analysis in a neoantigen prediction. So with all these challenges that I have laid out here, how can we make this neoantigen analysis from mission impossible to possible? So with that, I would like to transition to Vladimir and let him share with you our successful story in the neoantigen discovery and to showcase our new neoantigen workflow, which is by far one of the most complex workflows living on the platform. So with that, Vladimir, I will let you take it from here.
Thank you, Xavier, for the wonderful introduction, and special thanks to Jen and Abby for organizing this webinar and giving us the opportunity to show last 3 years of our work that we did on discovering neoantigens. So before digging into the details about the flow itself and the methods being used, I would like to give you a short introduction about the cancer immunotherapy in order for you to better understand the significance of accurate neoantigen prediction and discovery. So first traces of immunotherapy date back to 25 centuries BC, when one of the Egyptian pharaohs used poultice with incision to treat some type of cancer, and he managed to obtain some small regression of it. And only 45 centuries after that, the proposition for using cancer immunotherapy as a treatment appeared for the first time in Show House, when Dr. Chase was supporting it as a treatment to one of his patients, the therapy which would train the immune system to recognize and destroy cancerous cells and leave the healthy cells alone. Not that much that the screenwriters of the house were prophets, but the large advances in sequencing technology enabled huge growth in cancer immunotherapy, which largely relies on this area. And we can surely say that nowadays we live in a golden era of cancer immunotherapy. So first, the general definition, something what Dr. Chase said on the previous slide. So any artificial stimulation of the immune system that can be used to treat cancer. There is a rough division of cancer immunotherapies into these three groups. So most of them fall into one of them. First is includes checkpoint blockades, inducing tumor cell destruction. They target special receptors on surface of the cell used to restrain immune reaction. The third group is consisted of programming immune cells, so identifying first potential neoantigens and extracting T cells from the patient and returning them back to the patient. And the second group is consisted also on identifying potential neoantigens and creating vaccines from them and then administer them back to the patient. So far, there are 18 approved gene therapy products, and some of them is in this group is this viral therapy also, which is different from these three, and it's consisted of injecting virus into melanoma, and where it multiplies and destroys directly cancer cells. So next few slides we'll go a little more deeply into each one of these groups. So checkpoint blockade targets usually PD-1 or CTLA-4 receptors on cells, which are used to restrain and better control the intensity of the immune response. unfortunately, some types of cancer exploit this mechanism and grow their own PD-L1, for example, receptors, as you can see on the first image. And when these receptors bind to the T cell, they deactivated it. And even when T cell recognized the neoantigen on the surface, it cannot start the apoptosis pathway and destruction of the cell because it's being deactivated. So these drugs, in the form of antibodies, they administer to the patient, and they bind to each of these receptors, and they enable activation of the T cells when neoantigen sequence is recognized. So it's not personalized therapy. It can be used on in many patients, and it targeted malignancies with a large mutational burden. And last couple of years, it's also been used with many other types of cancer, such as melanoma, lung cell, kidney, bladder, Hodgkin's lymphoma, and ovarian cancer. Also, it's very important what can be seen from the Nobel Prize in 2018, which was given for independent work of two scientists working exactly on checkpoint blockade-based drugs and treatments. So next group is consisted of programming immune system cells, and first step in it, as you can see on the image on the right, is consisted of removing T cells from the patient's blood and changing their receptors with special chimeric antigen receptors, which recognize and target exactly neoantigen, which is present in a certain type of cancer. After that, the modified cells are multiplied several million times and administered back to the patient in several phases. There are a couple of FDA-approved drugs based with this therapy. First one is for non-Hodgkin's lymphoma, and second one is for B-cell lymphoma. And the third group is consisted of administering neoantigens directly to the patient. But first, let's see the process of how neoantigens end up at the surface of the cell. So every cell in our body has mechanisms to cleave endogenously synthesized proteins in proteasomes and transfer these fragments with special transport mechanisms through endoplasmatic reticulum and Golgi apparatus into the surface of the cell, where they, with certain probability, bind to MHC, forming to major histocompatibility complex molecules, protein, forming MHC complex. This complex is expressed at the surface of the cell, presented to the cells of the immune system, and in a way it represents an indicator of what is present inside of the cells. So immune system cells, they scan every cell in our body like a police officers and check what's present inside, and if this fragment is not wild type protein usually present in the patient, if it is alternated due to somatic mutation, then it represents neoantigen, and it's present only on cancerous cells, and the job of the immune system is to destroy these cells and to save our organism. And this operation happens thousands of times every day. So this is on the image of the right, you can see the entire flow of the neoantigen therapy and the treatment. So first step is to perform sequencing, usually whole exome and transcriptome of the patient. So the sample is taken usually from the blood and from the tumor tissue. Then the second step is neoantigen discovery, bioinformatics analysis. This is what we do. This is what we will talk about in the rest of the webinar. And the output of this part is a list of prioritized and scored neoantigen candidates, which are first further validated in the laboratory with usually with flow cytometry, and then selected neoantigens are manufactured and delivered in several ways. One of the ways to deliver them is through unformulated micro RNA sequence, which encode this neoantigen, and it's usually administered directly to the lymph nodes of the patient, where it's being swallowed by immune system cells, macrophages or dendritic cells, and after swallowing them, the translation to the protein will occur, and these peptides, neoantigens, will appear on the surface of the dendritic cells, and the immune system will recognize them, and it will train T cells, which will, through bloodstream, circle the body and locate the tumor, which expresses the same neoantigens, and be able to destroy them. The second group is similar. It's similar delivery strategies, just the neoantigens peptides are directly administered. So the downside of these first two strategies is that a huge number of proteins, of peptides and microRNA is being degraded, and because it's unstable in the intercell environment. That's why this fourth group with the nanovaccines and biomaterials is many groups are working on it, and its main feature is that synthesized neoantigens proteins are more sustainable in this environment, so the immune system has more time to absorb it before they are degraded. And the third group, it's consisted of taking out from the patient's blood the dendritic cells and pulsing peptides directly, pulsing neoantigens directly to them, and when they present it on the surface, the cells are returned to the patient, so that way avoiding a huge possibility of neoantigens being destroyed in a patient's intercellular environment. And also it's important to mention that these therapies are almost always used in a combination with checkpoint blockade inhibitors. Still, a neoantigen cancer vaccine is a very hot area, also in science and research. Currently, there are 4,400 mentions in papers since 2019, and a little less than 1,500 only in 2020. In this table, you can see some of the research is being conducted. Some of them in clinical trials, in preclinical studies. Some of them are in phase 1 or phase 2 studies, and they include several different types of cancer. Similarly, with checkpoint blockade, first it's been used with the tumors with large mutational burden, but the last couple of years it's been also used with other types of tumor which has much smaller mutation burdens, such as glioblastoma. And it's important to mention some downsides of the immune therapy. First is the possibility of developing autoimmune response or disease. This is especially present in a CAR T cells therapy. So it's very important to investigate very thoroughly the immunogenicity of the neoantigen candidates being administered, and make sure that they are not present in other tissues of the body, where because they can be attacked by patients' immune system. And still, unfortunately, this kind of treatment is very expensive and still not available to the majority of the population, but it's getting better together with together with fall of the price of sequencing technology. Also, the price for manufacturing peptides and vaccines will go down. So we hope in next couple of years or decade will become available to the majority of the population. So we'll go to neoantigen discovery workflow, and show you this bioinformatics analysis that can discover and prioritize neoantigens from the patient. So this workflow represents a very high-level image representation of neoantigen discovery. It has several processing units. On input, it receives raw sequenced reads from both normal and tumor tissue, together with the RNA, and it performs germline and somatic variant calling, which includes reconstructing of the patient's DNA sequence. It is very important to perform this in order to have accurate neoantigen candidates, and usually this reconstructed is done using reference genome and information that we share the majority of, among humans, share the majority of the DNA. So we have something like 99.6 same DNA code, and the output of this part is filtered variants, which are using our custom scripts or scripts for extracting proteins and creating neoantigen candidates. And the last step is it's consisted of scoring of each neoantigen candidate and outputting the list of prioritized candidates. And this is how the workflow looks like on SevenBridges platform. It is same as in previous images. It's consisted of several processing units. So each one of these green circles is a sub workflow, and consisted of other embedded bioinformatics tools. The gray ones are inputs, and the outputs are on the right. As Xavier mentioned, same as every workflow on SevenBridges platform, it's fully described in a common workflow language, making it portable and reproducible. It is the most complex workflow. The platform consisted of total 130 bioinformatic tools, and it executes in 7 to 14 hours, depending on the mutational burden and the cancer type. Just as an illustration, if we didn't put the tools into sub workflows, this is how it would look like with all the tools on one canvas. So it's a little messy. Of course, we don't want to have it like this without logical units. So this diagram shows a little more detailed representation of the processing inside neoantigen discovery workflow. As you remember, the inputs are sequenced reads from tumor and normal in FASTQ format, and after the alignment step to aligning the reads to the reference genome and HLA typing, we perform somatic variant calling, variant filtering, then phasing of the variants. Then variants are dividing into two groups, where each group belongs to the same haplotype. And then we perform translation to the proteins. And when we create neoantigen candidates, we score them by predicting MHC binding. It's a probability that certain neoantigen candidate will appear on the surface of the cell. And then all the results together with RNA data is fed to this analyze epitopes tool, which aggregates all the data and outputs scored neoantigen candidates. So I suppose this diagram is pretty complicated, especially if you are not that much into bioinformatics. That's why we decided to divide it into several processing phases, which we will show on next couple of slides. So this here is just an illustration how the entire flow works, and the next couple of slides will go in more details through every unit. So the first part of the workflow is consisted of DNA calling and HLA typing. So for tumor and normal reads, we perform alignment with BWMM and we perform HLA typing from the tumor sample, and then aligned reads from both tumor and normal tissue are fed to somatic variant calling. There we use two tools for calling. One is the Mutec2 and second one is Strelka2. And we also perform germline variant calling with the GATK4 haplotype caller in order to call the germline variants. So for accurate reconstruction of the genomic sequence, we need to have both germline and somatic variants. Then, as a final step of this stage, we perform optional filtering of DNA variants, where we can optionally remove some of the variants that are marked as low quality within these two callers, and we can also select whether to use union or intersect of these two somatic callers. So if our analysis requires large recall, if we want to cover all possible neoantigen candidates, then we will select union here. And if we are more interested in precision, then we want to target, for example, only variants that are being called by both somatic callers. So we would select intersect here. And all germline and somatic variants are passed to the output of this section. Next, in parallel with that, we perform RNA expression and variant calling. For RNA expression, we use Salmon workflow. We also have support for RSAM, and for RNA variant calling, we did it according to GATK best practices for RNA-seq. So it starts with the star aligner, and it includes split and cigar reads, base recalibration, and the haplotype caller. The output of this part is RNA variants and the RNA expression table, where for every transcript inside the gene annotation, from the gene annotation file, we give a score of transcripts per million that represent its expression. And next part is consisted of, let's say, translating or applying variants to the reference genome and extracting neoantigen candidates. And this illustration shows this very complex process in a, let's say, less complex manner. You can see a couple of variants that fall on certain positions of the genome. For example, a variant that falls into intron sequence, it's currently being ignored because it cannot make alternation in a protein sequence. And for example, this variant, which is somatic and which falls into coding region, it makes changes in the reference genome sequence, and after transcribing it to RNA and translating it to protein, it makes a change from the reference genome into one amino acid. Let's call it altered amino acid. And if we extract this part of the protein, of the patient's protein, around this altered amino acid with some area, it's default 10 amino acids from each side, but it's also configurable for every analysis. This protein, it's called neoantigen nest. Why it's called neoantigen nest? Because it gives birth to the neoepitopes, as you can see next. So neoepitopes are sequences in size from, let's say, 8 to 15 amino acids, and they include this altered amino acid, as you can see here. So, and the final step is to assign each of these neoepitopes HLA type, and this pair, so neoepitope plus HLA type, it creates neoantigen candidate. So this is neoantigen candidate. This combination of the neoepitope, altered peptide sequence by somatic mutation, and HLA type. And this is how is this protein extraction and prioritization done inside the workflow. So on the input, we receive filtered DNA variants and HLA types, and together with the RNA expression and the RNA variants. So first, here we perform phasing of the variants. On the next slide, we'll show the simple example of variant phasing, so you will understand better its significance. And then after this stage, the variants are divided into two groups, where each group contains variants that are present on the same haplotype. We call it maternal and paternal. And each part creates their own neoantigen candidates. And for every neoantigen candidate, we perform MHC binding prediction. It's a process which makes scoring of the neoantigen candidates by giving them probability that they will appear on the surface of the cancer cells, and we do it with several currently available tools such as NetCTL-Pen, NetMHC-Pen, Pickpocket, MHC Nuggets, and scores for all of these tools together with the RNA expression and RNA variants are fed to this final aggregation to analyze epitopes, which performs additional optional filtering of the candidates, such as removal of the wild type proteins. For example, it can happen that some of the neoantigen candidates, some of the protein sequences is present in wild type protein database, which is an input, another input for this tool. And we can optionally remove this. By default, they are removed, but can be turned off if necessary. And the second example of filtering is that we can remove all neoantigen candidates that originate from variants that are not called with RNA. And we can set, for example, the binding weight. So at the end, when we give the final score of the neoantigen candidate, we can decide whether to trust more, the binding prediction score from some tool or RNA expression. Also, we take into account RNA variants. So why we do that? Because with the RNA expression, we receive results about expression for a certain transcript, but we don't have information whether this transcript is, whether this allele is reference or alternate. So whether really the allele which contains this neoantigen mutation is expressed or the other one. So, but when the RNA variant is called, then we are certain that the expression of the alternate allele is really expressed. And as I said, this is a simple example of that shows significance of performing variant phasing. You can see here two heterozygous variants, which are very close one from the other. And when we perform, we want to perform the reconstruction of the nucleotide sequence from these two variants. So since they are close, we can form here four possibilities, and each of these four possibilities will code different protein. Thus, it will call different neoantigen candidate. But as a common fact, we are diploid organisms. We don't have four haplotypes, so two must be wrong. And how could we know which two are correct and which two are wrong? We can know that only with the information about phasing of the variant. So if this information is present, so we have a special tool that performs phasing of the variants. So if this information is present, we can know exactly which variants go together and which don't go together, and eliminate these false two peptides and keep only two that are correct. And the final outcome of the workflow is this set of scored and prioritized neoantigen candidates together with a huge number of information that could benefit to additional filtering or giving more or less trust to it. On the right side, you can see this animation showing this final outcome on the workflow on our platform. And on the bottom side, you can see some of the fields from the table with the neoantigen candidates. First group gives basic info, so the peptide sequence, HLA type, information about the somatic variant. Then next group, it's consisted of scores from several binding prediction tools that I mentioned. Also information whether this peptide is present in wild-type protein database. And we also calculate agretopicity, which is very important parameter. It shows the difference in a binding prediction score between alternate allele and the reference allele for this neoantigen candidate. And it's important to have it larger than 0, or sometimes larger than 0.1 or 0.2. It gives more credibility to the neoantigen candidate. And some papers are considering agretopicity as a criteria that candidate is a neoantigen candidate. Only if it has agretopicity larger than 0, it's called neoantigen candidate. And we also provide additional information about the somatic variant consisted of variant allele frequency. It boosts its reliability, and both tumor variant allele frequency, which should be larger than at least 0.1, 0.2, and also normal variant allele frequency, which should be close to 0. And the last group consisted of RNA parameters, so the list of isoforms of the gene on which the mutation in neoantigen candidate falls to, transcripts per million score, whether this variant is called in RNA, and also expression and genotype of both reference and alternate alleles. And this is an example of the analysis of the task of neoantigen workflow on SevenBridges platform. On the left side, you can see reference files, inputs, all necessary databases, reference genomes, index files, gene annotation files, and you can see row reads. They are given all on the same input in order to allow batch processing of more than one sample. So this workflow has capabilities to process dozens or hundreds of samples in only few clicks. So it will spawn dozen or 100 instances for each sample and run the workflow and provide the result. And on the right side, you can see some output FASTA files, VCF files with called variants, and of course merge the epitopes, merge neoantigen candidates, the table that was shown on one of the previous slides, together with some information about the number of variants, of coding variants, and etc. And as Xavier mentioned, the big challenge with this type of analysis is validation. Currently, there is no publicly available truth set or best practices workflow, which is why this Tesla consortium was found by Parker Institute. The acronym is Tumor Neoantigen Selection Alliance. It gathers more than 35 leading neoantigen research groups from both academia and industry, and they organize challenges in which everybody is welcome to participate, and they provide validated sets for more than 30 neoantigen candidates. They provide flow cytometry validated neoantigen candidates, so the participants can test their workflows with them. And SevenBridges participated in 3 rounds, round 2X and 3 of this neoantigen challenge, and our workflow managed to detect and assign very high scores to the majority of the flow cytometry confirmed neoantigen candidates. The detailed results about this challenge will be published soon by Tesla organizers, and also we are preparing our own paper, which will describe this workflow, its capabilities and features. Also, it's important to mention one more time the Tesla goals were to create best practice neoantigen workflow and the truth set. Last, to summarize a bit, I always want to point out these two important things distinguishers for our neoantigen workflow comparing to the other publicly available solution. The first is that we perform integral neoantigen analysis described in a common workflow language, so from FASTQ file to prioritized and scored with many details neoantigen candidates with many optional filterings and prioritizations. And the second advantage is that we perform and take into account the wide range of mutational events. This is implemented in our custom scripts for protein extraction. And these changes include consider both germline and somatic variants, both SNPs and indels. Neighboring SNPs and indels are also taken into consideration, variants of stop codon, phasing of the variants, and customizable transcript selection file. And this is also one of the key elements that we plan to publish in our papers to calculate the importance of each one of these mutational events and to calculate exactly how many neoantigen candidates is lost if we don't take into account one of these mutational events. Thank you very much for your patience, and hope to hear feedback from you.
Thank you, Vladimir, for the fantastic talk. So now we are open for Q&A. If we cannot answer all the questions due to time constraint, we'll respond the rest after the webinar. So please do not hesitate to post any questions in the chat box below. Okay, so we have the first question here. So it's really impressive to see your workflow can capture a variety of mutational events. So with that, have you also considered to predict the immunogenicity of neoantigen candidates?
Oh, thank you. It's a great question. It is one of the final step of the workflow. So to mention one more time, we predicted the possibility of neoantigen candidate to appear on the surface of the cancerous cell. But the final step is to predict immunogenicity and the probability that this candidate will actually bind to the T cells that are tumor infiltrating lymphocytes that are present there. And with that, for that prediction, we require additional data for sequenced T cell receptors of the cells in the tumor environment. And it can easily be added to the workflow. There are a couple of tools that currently can perform this type of prediction. One of them is NetTCR, and it's on our list as one of the next steps for adding it to the workflow. And important to add, similarly like with the other neoantigen and validated neoantigen data, it's really hard to find publicly available data that would contain raw reads from both tumor and normal, and also sequence the T cell receptors in order to test the entire flow.
Okay. The next question is it possible to perform the prediction of neoantigen candidates if there's no RNA-seq data available for the patient? So in other words, you only have the tumor normal DNA.
Yes, it is possible. The last tool in the workflow that performs aggregation of results, it is set in a way that RNA data is optional, so it will be ignored. Like it's not there. So the candidates will be prioritized only with the MHC binding scores from the selected tool, and they will be scored. So it is possible. Of course, it's important to mention without RNA, the predictions are not that much reliable, because we can have candidates with a very huge MHC binding score, but they are not really expressed in the RNAs. Thus, the proteins from these transcripts from these nucleotides are really not present in the patient, in the cancer cells, and not expressed at the surface.
We have another question. Says, do you run analysis on whole genome, or do you restrict it to chromosome 6 when where the MHC is encoded?
It is possible to run it also on whole genome, and we don't run it only on MH6. So it's the entire exome of the patient. All the genes are processed, and we consider only chromosome 6 for HLA typing. So neoantigens from all genes are tested and taken into account. If whole genome sample is provided, since neoantigens are extracted only from genes, we will, for now, the intron regions and regions outside of the genes will be ignored.
So there's a question asking, it's very impressive to see like the Tesla challenges. So if we've gone through and all the populations once gone through, what's the consequence, what's the impact, and what the end users would expect to benefit from these publications?
Yeah, the impact of this challenge and the results, it's really huge, because not only they will publish the results of the workflows from the participants, but they also plan to publish all the test data. So all data that was used in a challenge, they plan to publish it at some point, making it available to anyone, to research group laboratories, so the researchers can build their own prediction tools, can test their own workflows, can tune them. They can also, the community can, with the help of Tesla Consortium, come out at some point with the neoantigen best practices workflow. And also, since it's very complex and there are so many parameters of the workflow, the community can come out with optimal thresholds or optimal values for some parameters. And of course, it's very important to mention why the accurate prediction of neoantigen is a must-have. So, as mentioned during the talk about cancer immunotherapy, it is very important that these synthesized neoantigens are really the proteins that are expressed at the surface of the cancerous cells. So, if an error happens, some wild-type protein can be synthesized, and it can provoke autoimmune reactions. So, it's very sensitive data, and the validation for every step needs to be performed very thoroughly.
So I think we have time for the last question. Okay. So the last question is, how does tumor heterogeneity affect the selection of neoantigens?
This workload does not consider tumor heterogeneity for now. So we only have one tumor sample in the workflow, and we calculate all the variants. And of course, it's important to include it, especially because of the cancer immunoediting. There are several researches that target this this effect. So it can happen that clones of the tumor they stop expressing some of the neoantigen candidates that were predicted in the primary tumor or in one of the first clones of the tumor, and that points out that prediction of neoantigen candidates it's not sufficient to do it only once. So it should be done several times as the time passes by during the treatment, because some of the neoantigen candidates can be lost in the patient, and maybe some good or strong neoantigen candidates can appear.
Okay, thank you so much, Vlad. I think that's all the time we have today. Once again, we really appreciate your participation and all the good questions that you have posted here. And we'll also respond to the rest after this webinar. So please do not hesitate to leave any comments or questions in the chat box. And with that, we do look forward to seeing you again in our upcoming webinars. Thank you all, and have a good one.
Thank you.